Well, it's time to begin our discussion on neoplasia. I will conclude this topic with treatment of cancer. The go-to standard is surgery, but there are other interesting treatments to discuss as well. The question is, are we winning the war against cancer? I always joke with my traditional face-to-face students that if we were mice, we would have the cure to most cancers already. Scientists have developed, discovered drugs and treatments that cure different cancers in mice, but for whatever reason, their effectiveness does not transcend into humans. But we are still winning that war against cancer. Every day we are developing new forms of chemotherapy or biotherapy. Cancer is not the death sentence it once was. The 5-year survival rate has improved drastically since the 1960s. Different types of cancer have different pathogeneses. Cancer is not a single disease. Let's start off simple. What is the difference between dysplasia versus neoplasia? Well, I'll remind you from our earlier lectures that dysplasia involves cells that have changed in size, shape, and organization. Hyperplasia involves abnormal proliferation of cells, but should stop once the quote-unquote stressor or causative agent is removed. Neoplasia differs from the other two because this now involves disorder of altered cell differentiation and growth. The new growth tends to be crab-like because cancers grow and spread by sending crab-like projections into the surrounding tissues. So neoplasia is an abnormality of cell growth and multiplication, and it is characterized by excessive cellular proliferation, even after the stimulus is gone. It is uncoordinated growth and tissue infiltration. At the molecular level, there are disruptions of the growth regulatory genes, and all of this develops in a multi-step fashion. As I continue to lecture, pay attention for these regulatory genes when we address the cell cycle. It is time to address the cell cycle, or the interval between each cell division. The cell exists in a phase known as interphase, the longest. Interphase has 3 subcategories, G1, S, and G2. The cell cycle begins with G1, which stands for gap 1. Cellular activities during G1 involve preparations for DNA replication, which will occur during S phase. There is increased protein synthesis and synthesis of organelles and cytoskeletal elements. The S phase stands for synthesis phase and can last 10 to 12 hours. During this precarious time, the chromosomes are being replicated. Each strand is unwound by a helicase, and each strand serves as a template for a new daughter strand. This process is referred to as semi-conservative replication. After S phase, there is a second gap phase, G2. This is a quote-unquote pre-mitotic phase. Enzymes that are needed for cell division are synthesized. This involves gene expression. The DNA is checked for errors and damage that might have occurred during replication. M phase is the time when the nuclear DNA is divided between 2 daughter cells and also has a component known as cytokinesis, when the cytoplasmic contents are split between 2 daughter cells. Perhaps you are recalling terms involved in mitosis like prophase, metaphase, anaphase, telophase, and lastly, cytokinesis. In some cells, they may enter a special phase known as G0 or gap 0. I always learned it in grad school as G0. In this case, a cell leaves the cell cycle temporarily or permanently. It exits the cycle at G1 and is often called quiescent, but that implies that the cell is inactive and that is most certainly not the case. The cells are busy carrying out their functions in the organism. Often, G0 cells are terminally differentiated. They will never re-enter the cell cycle, but instead will carry out their function in the organism until they die. For example, neurons. For other cells, G0 or G0 can be followed by re-entry into the cell cycle. Most of the lymphocytes in human blood are in G0. However, with proper stimulation, such as encountering the appropriate antigen, they can be stimulated to re-enter the cell cycle at G1 and proceed on to new rounds of alternating S phases and mitosis. G0 represents not simply the absence of signals for mitosis, but an active repression of the genes needed for mitosis. Cancer cells cannot enter G0 and are destined to repeat the cell cycle indefinitely. Before the cell moves from one phase to the next, there are checkpoints. For example, there is a checkpoint between G1 and S phase, known as the G1S checkpoint, sometimes referred to as restriction point 1. And there is one between G2 and mitosis known as the G2M checkpoint, or restriction point 2. At G1S, the checkpoint involves proteins that check the DNA for any damage that should be repaired prior to replication. For example, uracil is found in DNA. It can result from spontaneous deamination of cytosine. This uracil, if not repaired, can cause a point mutation when the DNA is replicated. The second checkpoint will check for any DNA that wasn't replicated, and it will also be checked for any replication errors. In the next few slides, I will identify proteins called cyclins. There are many types, and these are expressed at specific times of the cell cycle. Cyclins control the entry and progression of the cell through the various phases and do this by interacting with CDKs, cyclin-dependent kinases, to activate them. A kinase is an enzyme that attaches a phosphate group to other proteins. Proteins that are phosphorylated will change their activity. CDKs, unlike cyclins, are constitutively expressed. They are in the cell all the time. The idea is they are not activated until their cyclin partner is expressed. We will also see antagonistic proteins called CDK inhibitors. Really, it all comes down to maintaining a series of checks and balances to ensure proper cell growth and division. Let's go through the cell cycle again, but this time let's focus on the cyclins and CDKs and CDK inhibitors I introduced on the previous slide. G1 cyclins involve D cyclins, not shown in your book. The S-phase cyclins, E and A, and mitotic cyclins, B cyclins. Again, their levels rise and fall with the stages of the cell cycle. CDKs include G1 CDK, known as CDK4, S-phase CDK, CDK2, and M-phase CDK, known as CDK1. Their levels in the cell remain fairly stable, but each must bind to the appropriate cyclin, whose levels fluctuate in order to be activated. The CDKs add phosphate groups to a variety of protein substrates that control processes in the cell cycle. During G1, a rising level of D cyclins bind to CDK4 and signal the cell to prepare the chromosomes for replication. A rising level of S-phase promoting factor, which is a complex of A cyclins bound to CDK2, enters the nucleus and prepares the cell to duplicate its DNA. As DNA replication continues, cyclin E is destroyed and the levels of mitotic cyclins begin to rise in G2. The B cyclins combine with CDK1 to form M-phase promoting factor, or MPF, and this initiates the assembly of the mitotic spindle, breakdown of the nuclear envelope, cessation of gene expression, and condensation of the chromosomes. The two pictures on the screen show how these cyclin and CDK complexes form and are subsequently degraded. The cyclin CDK complexes are broken down by the CDK inhibitors. So it really is the formation and accumulation of the cyclin CDK complexes and their subsequent removal by CDK inhibitors that regulate the cell moving from one phase to the next. And you should keep in mind that new cyclins and CDK and even CDK inhibitors are being discovered. The cell cycle story is continuously changing as we discover more of the chemical signals that regulate it. I like this last picture. It focuses on cyclin B and the synthesis of MPF. And in this graphic picture, you see levels of cyclin B and MPF rise in a coordinated manner to promote mitosis, and then their levels drastically decline as the cell enters interphase, only to lead to the accumulation of cyclin and MPF as the cell approaches mitosis again. It is important to note that these cyclins, CDKs, and CDK inhibitors are proteins. Mutations in their genes have been associated with cancer. These genes are considered to be proto-oncogenes, and I'll speak more on this later. This should not be surprising since checkpoint failures would allow the cell to continue to the next phase despite damage or lack of integrity of macromolecules. A perfect example of a failed checkpoint can be seen in the p53 protein. p53 protein, of course, is the product of the p53 gene. p53 is responsible for checking DNA integrity before DNA replication and can halt progression of the cell cycle in G1 by blocking the activity of CDK2. Both copies of the p53 gene must be mutated in order for this checkpoint to fail. So mutations in p53 are considered to be recessive. That is, both copies must be mutated. And p53 is really seen as a tumor suppressor gene. We will have more discussion on tumor suppressor genes later. Your textbook has many pages dedicated to cell proliferation and cell differentiation. Cell differentiation is certainly one of my favorite topics, but I should also discuss cell proliferation. In your body, you have about 200 different types of cells. These can be divided into three large groups. 1. Well-differentiated cells like neurons and skeletal muscle that rarely divide and reproduce. 2. The progenitor or parent cells that continue to divide and proliferate, like blood cells, skin cells, etc. 3. The undifferentiated stem cells that can enter the cell cycle and produce the progenitor cells in the body when the need arises. As a stem cell replicates and divides, it will give birth to two daughter cells shown here in the bottom picture. One of the two daughter cells will remain as the stem cell, while the other will become a progenitor cell, which can also replicate and divide to give rise to daughter cells. Each of these daughter cells is becoming increasingly more determined. That means there is selective gene expression, some genes turned on, others kept off. The end result, after a series of maturation steps, the cells will become so specialized that they will lose their mitotic abilities. The steps required for a primary tumor to metastasize are the following. First, you need the primary neoplasm to replicate and divide. That tumor must be able to break through the extracellular matrix. Cancerous cells do this largely by expressing the gene for the type 4 collagenase enzyme, and this allows them to break through the basement membrane of a nearby capillary. This leads to intravasation of cancer cells into the capillary, and evasion of immune cells that could destroy them. Many cancer cells will form aggregates with thrombocytes, basically coating themselves with platelets to help with immune system avoidance. Then the tumor cells need to be able to break through a vessel again and take up residence in an area where the microenvironment will support them in a process called extravasation. And of course, those cancer cells need angiogenesis. We all have areas in our body where we have developed tumors. The thing is, they eventually stop growing and are contained because they don't have enough nutrient supply. Read, they don't have a blood supply to deliver nutrients. It's when the cells switch their chemical signaling that triggers angiogenesis that the tumor can now get big enough to be detected. Three primary things will now dictate the rate of tumor growth: number of dividing cells, length of the cell cycle. If shorter, it can double its population size faster, and the number of cells being lost, if any. The growth fraction of tumor cells is greater. Growth fraction refers to the ratio of dividing cells to cells in G0. Cancer cells don't necessarily have a shorter cell cycle. Their rate of growth is usually due to more cells actively dividing rather than going into G0. When we speak of doubling time, this refers to the length of time to double the mass of a neoplasm. In order for a tumor to be detectable, it needs roughly 30 doublings, or needs to be about 1 billion cells big. Let's start to focus now on known causes of cancer. Your textbook separates causes of cancer into two broad categories: genetic and host/environmental factors. We will begin with a focus on the genetic approach to cancer. A proto-oncogene is a normal gene that is found in the nuclear DNA. These genes code for proteins that would promote cell growth and division. These proteins might include growth factors, or receptors for growth factors, or second messenger factors that alter gene transcription and translation of other proteins so that a cell can increase its growth before division. In this picture at the top right, you are seeing a picture of a growth factor binding to a receptor and activating a second messenger system within the cell. Second messengers activate kinases, proteins that phosphorylate other proteins. The example here being mitogen activating protein kinase. And this MAP kinase is shown phosphorylating a protein involved in gene expression and thus turning on genes that are involved in upregulating mitosis, and that means more cell division. When proto-oncogenes have a mutation that leads to a quote-unquote gain of function, then these genes are overly expressed, and we now call this gene an oncogene, or a gene that promotes cancerous growth. In some cases, the mutation leads to a new protein altogether that leads to aberrant cell growth. In contrast, tumor suppressor genes code for proteins that help downregulate the cell cycle. The two most well-known genes are the retinoblastoma gene, RB, and the p53 gene. These two proteins participate in cell checkpoints. When tumor suppressor genes are mutated, we are worried about the loss of their function, or more simply, the loss of cell checkpoints to arrest the cell cycle to allow for DNA repair. So I'm hoping you can see the balance between the activity levels of these two types of genes. In a normal cell, they should balance each other. Cancer can occur due to activation of growth-promoting oncogenes, as shown in the picture, or conversely, it can result from inactivation of tumor suppressor genes, or in genes that control apoptosis. All can lead to unregulated cell differentiation and growth, which leads to a malignant neoplasm. First, it is important to remember from your text and from the videos I provide to you this week that cancer is not a single disease and it does not arise from a single hit. A normal cell sustains many insults or exposure to damaging agents like chemicals, radiation, viruses. And most of the time, these insults cause DNA damage. Most of the time, the cell responds in an appropriate manner, either repairing the damage, going into senescence, or apoptosis. It's when the cell receives too much damage that the protective/corrective mechanisms are overwhelmed and we start to see malignant neoplasms arise. So it is all about initiation of DNA damage by some physical agent that overwhelms a cell's corrective mechanisms. DNA damage leads to DNA mutations, a fixed heritable change, meaning it now is fixed in place in the genetic code, and this change can be transmitted to future daughter cells. There is subsequent further genetic insult in the activation of oncogenes, loss of tumor suppressor genes, etc., in what is called the promotion phase. And when a neoplasm recruits a blood supply, has uncontrolled growth, has invasiveness through the basement membrane, and lacks differentiation, that the progression stage of cancer occurs. Now, how does a proto-oncogene get activated or mutated into an oncogene? There are several different types of DNA mutations: a point mutation, translocation, or even a duplication/amplification of a gene, even insertion of viral DNA that leads to increased expression of a proto-oncogene. You might be wondering how a gene could be duplicated or amplified. Well, that's usually from viral infection. Some viruses insert their DNA into our genome, and when their packaging machinery clips their DNA back out of our DNA, sometimes they pick up a gene from our genome, and this can be reinserted into another cell, and now that other cell would have an extra copy of that gene. An example of an amplified gene that leads to cancerous growth is human epidermal growth factor receptor 2, HER2. This gene is amplified in about 30% of breast cancers, and its overexpression leads to a poor prognosis due to the aggressive nature of this neoplasm. A classic example for a point mutation generating an oncogene is the story behind the RAS oncogene. It's really not just one oncogene, it's a family of oncogenes. These RAS proto-oncogenes normally act as second messenger systems that send growth signals to the nucleus to increase cell mitotic rate. When a point mutation occurs in these genes, it leads to a gain of function, and now they are oncogenes that promote too much cellular division. What about translocations? Well, some chromosomal ends are more sticky than others, or are more prone to double-strand breaks. As such, you can have two chromosomes trying to seal their ends by binding with each other. If it's a reciprocal translocation, a person may be quote-unquote lucky in that they still have the normal number of chromosomes in their cells and there is no disruption of genes where the chromosomes split and reunited. But sometimes this reciprocal translocation can disrupt a gene or maybe even create a fused gene in the splice site where the gene is now in close proximity to a different promoter that leads to excessive expression of the gene. Now I'd like to tell you about the translocation that occurs to form the Philadelphia chromosome. In most cases of chronic myelogenous leukemia, CML, as well as some cases of acute lymphocytic and myelogenous leukemia, ALL and AML, a piece of chromosome 9 and a piece of chromosome 22 break off and switch places with each other. This new chromosome, which is mostly chromosome 22 with a piece of chromosome 9 stuck to it, is called the Philadelphia chromosome, designated here at the right of the picture as PH superscript 1. So now chromosome 9 gets longer and chromosome 22 gets shorter due to the swapping of DNA between the two chromosomes. The Philadelphia chromosome is only found in the affected blood cells. Because of the damage to the DNA, the Philadelphia chromosome results in the production of an abnormal enzyme called tyrosine kinase. Tyrosine kinase is a huge player in one type of second messenger cascades and sends growth signals to the nucleus. So, just to review, proto-oncogenes code for proteins that promote cell growth and division. They can be growth factors, the example here being IGF, insulin-like growth factor. They can be receptors for growth factors like human epidermal growth factor receptor, HER2, or signal transduction factors like a cytoplasmic kinase, the example from the previous slide being tyrosine kinase. The ultimate target being DNA and transcription of proteins that are involved in the cell cycle. Shown here is cyclin D. Shown on this slide, a mutation in a proto-oncogene leads to a gain of function, and this oncogene can code for more increased growth factor, in picture 1, more growth receptors, increased second messenger system activity, and all of this leads to an increase in gene transcription, expression of genes that code for cell cycle proteins, like cyclins. Let's switch now to tumor suppressor genes. Tumor suppressor genes code for proteins that inhibit cell growth and division. They allow for the timing of our cell cycle checkpoints. The classic examples are P53 and RB. A picture of both is seen here. The gene for P53 is sometimes called TP53, or we use italics when we write P53 when we speak of the gene. P53 is a protein that halts the cell cycle and allows for DNA damage assessment prior to DNA replication during the S phase. Looking at the bottom picture, we see a normal P53 arresting the cell cycle and allowing DNA repair enzymes to repair DNA damage. If there is too much damage, we see P53 inducing apoptosis. Now, how can we get an abnormal P53 or RB protein product? Turns out these genes use typical Mendelian genetics. By that I mean you can be homozygous for 2 normal copies and you would have normal P53 or RB proteins. You can also be heterozygous for a normal P53 or RB gene and you would still make enough of those proteins to regulate the cell cycle. However, if you lose heterozygosity, LOH, and now have 2 mutated copies of these genes, you now fail to make P53 or RB, and now you lose your checkpoints. In other words, you need a 2-hit mechanism before these systems fail. The first damaged copy can be inherited, as shown here at the top picture. This is demonstrating inherited retinoblastoma versus sporadic retinoblastoma, where both copies of the RB gene are mutated by some agent at different times. The end result is if a cell lacks these proteins, then it fails to stop cell division. The cell continues to divide without repair to damaged DNA, and the damaged cells continue to divide. The genome becomes progressively unstable, and eventually the cell turns cancerous. Viral oncogenes are responsible for oncogenesis, resulting from persistent virus infection. Although different human tumor viruses express different viral oncogenes and induce different tumors, their oncoproteins often target similar sets of cellular tumor suppressors or signal pathways to immortalize and/or transform infected cells. In 1909, a farmer brought Dr. Francis Peyton Roos, a junior faculty member then at Rockefeller University, a hen that had a breast tumor. Roos performed an autopsy, extracted tumor cells, and injected the cells into other hens, which then developed sarcoma. This was the first experimental proof of an infectious etiologic agent of cancer, and the chicken sarcoma-inducing RNA virus was subsequently named the Roos sarcoma virus. After a half-century debate on whether viruses truly can cause cancer, Roos was eventually awarded the Nobel Prize in Medicine and Physiology in 1966 for his discovery of tumor-inducing viruses. It is now estimated that 20 to 25% of human cancers worldwide have a known viral etiology. Since that time, demonstration in 1980 of the first human retrovirus, human T-cell leukemia virus type 1, or HTLV-1, which causes adult T-cell leukemia, was a landmark achievement. Viral strategies for manipulating the expression of cellular genes to enhance viral persistence, viral latency, and survival of infected cells have provided numerous clues to the mechanisms of gene expression and their dysregulation during tumor development. There appears to be an epigenomic component to the development of cancer. In this hypothesis, there isn't a mutation that is occurring to the gene, rather external environmental influences cause changes in gene expression. In this picture, you are seeing chromatin, and chromatin is DNA wound up around histone proteins. The DNA must be unwound from these proteins for gene expression to occur. External environmental influences, drugs, aging, development in utero, diet, etc., can cause changes in the histones and their methylation status, shown here as a change in a quote-unquote histone tail. If we apply this concept to our previous slides, it's not hard to understand how a neoplasm could generate through changes in methylation of histones that lead to increased gene expression of proto-oncogenes or decreases in expression of tumor suppressor genes. Patterns of gene expression can be altered in one individual, and epigenetic studies now suggest that this change in gene expression could be transmitted to future generations. So if your patient has cancer, what would you expect to see? What are some of the manifestations? First, we know a neoplasm that has metastasized is an aggressive neoplasm that has the ability to enter the bloodstream and take up residence somewhere else in the body and recruit blood flow to deliver nutrients and oxygen. These are very greedy cells. As such, your patient will present with wasting syndrome. Anorexia refers to reduced food intake resulting in weight loss. The person is not ingesting enough nutrients even for their normal cells. People with cancer oftentimes have a decreased appetite, but anorexia alone in cancer patients does not explain the profound wasting syndrome we see in the patients. Cachexia means the normal cells are starving because even if the person were to eat or get nutrients, the cancer cells are taking more than their share of the nutrients. In other words, the metabolic demands of both the cancer cells and normal cells are not being met by the nutrient intake. The cancer cells especially are in a hypermetabolic state. There is even altered metabolism of the nutrients by the cancerous cells, and all of these factors lead to significant weight loss. Lack of appropriate glucose for other tissues of the body, like the brain, can explain the appearance of sleep disorders, which leads to extreme fatigue in the patient. Lack of nutrients for immune cells can mean the patient becomes more susceptible to secondary infections. The tumor itself can cause organ failure in the primary site or even in secondary sites. Lack of nutrients for normal blood cell synthesis can lead to anemia, or the invasive nature of the cancer can cause hemorrhage outright. For example, colorectal cancer patients may have blood in their stool. Of course, the go-to for cancer treatment has long been, and still is, the surgical removal of the cancer. Obviously, some neoplasms are easier to get to than others, so this might not always be an option. Also, removing as much of the tumor as possible means a better prognosis. I was reading a paper last year that talked about the difficulty in removing brain tumors because obviously the surgeon would like to remove all of the tumor but not remove healthy and much needed neurons. This paper used a scorpion venom to bind to cancer cells, and when a black light was shown on it, the cells fluoresced. This let the surgeon see which cells were the cancerous cells and leave behind the healthy neurons. Radiation therapy can be used. These are high energy waves that disrupt the cancer growth. However, there needs to be some access for the radiation therapy, and not all neoplasms lend themselves to radiation therapy, or they might be resistant to it. For example, melanomas. The radiation waves induce so much DNA damage that the cancer cells cannot continue to replicate and divide. Radiation therapy can be used alone or as an adjuvant, which means along with some other type of therapy, for example, surgery. There are different types of chemotherapies, and your book lists many of them. The idea is the more selective the chemotherapy is in targeting the cancerous cells, the fewer side effects the patient will experience. Chemotherapy targets rapidly dividing cells, and this is the reason why hair follicular cells end up being targeted by the chemotherapeutic agent, and the patient loses their hair. Different chemotherapeutic agents have different receptors, and if the cancer lacks a receptor, then the chemotherapy is ineffective. This is part of the problem with treating triple-negative breast cancer because 3 receptors that could be targeted are not made by this cancer, and so now the patient has fewer chemotherapeutic agents to use against the cancer. Some types of immunotherapy include taking matched natural killer cells or T lymphocytes from people who have low incidence of cancer in their families and giving them to people with tumors. The idea being that these immune cells would attack the tumor cells. So many more examples are being used, but let's move on. Biological modifiers include cytokines or interferons, monoclonal antibodies, and blood growth factors. Interferons are proteins made by cells in response to a variety of cellular or viral stimuli. There are 3 major types: alpha, beta, and gamma, each group representing the receptors they bind to on their target cells. Interferons inhibit viral synthesis and tumor protein synthesis, and thus more cancer cells pushed into G0. Monoclonal antibodies are made by injecting a mouse with a cancer antigen. The mouse starts to make antibodies to that cancer antigen. We then sacrifice the mouse and harvest the immune cells from the spleen that are making the antibodies. Then we fuse the mouse spleen cells with tumor cells that continue to replicate in vitro. This creates a hybrid cell culture called a hybridoma. You then plate this hybridoma cell line so that only one cell gets into a dish, and then you spend weeks, months, years trying to find the one hybridoma cell line that is making the antibody against your cancer antigen. Why does it take so long? Well, of course, there are many B cells in the spleen, each making its own antibody. So you create thousands of hybridomas, but only one makes the antibody to the cancer antigen you injected into the mouse. These antibodies then can target the tumor cell in the patient and inactivate it in different ways. Hormone therapy can include delivering an agent that binds to a hormone receptor. Some cancers are supercharged from hormones, and thus blocking a hormone's receptor can block how rapidly the cancer grows. Tamoxifen works in this capacity by blocking the estrogen receptor and is used to treat some types of breast cancers. This concludes our discussion on neoplasms.